Yeah.
Yeah, so I just wanted to, I'll introduce Brandon. So Brandon is a man of many interests, very interdisciplinary. He's following a long tradition of physicists coming into neuroscience. But just as an example of his interdisciplinary interests, I noticed that he's stabilized his computer with 3 books on the bottom. One is Introduction to Algorithms, the famous course in licensing and RAS book. The top book is Statistical Physics, and these 2 scientific edifices are sandwiching a book on religion. So it's a very nice, actually unified way to show Brandon's various very interesting facets. So Brandon joined my lab from the Applied Physics Department, jumped into a bunch of interesting projects. You'll hear about them, ranging from the analysis of neural data to a science paper, contributing to a science paper that showed how you can drive percepts in a mouse through tiny perturbations, the requisite theory for understanding that, understanding spontaneous activity in neural circuits, connecting to avalanches and their structure across many different species. And also simultaneously, Brandon was quite brave in joining an international collaboration of research labs that were a part of the International Brain Lab that's trying to do neuroscience in a different way by having many labs try to standardize data collection, analysis, and computation across the world. Brandon very bravely joined that collaboration and got some interesting results. So very excited to hear your talk, and take it away.
Thank you, Surya. Today we'll be exploring the many scales of neural activity underlying perception and decision-making. And I want to start by jumping into a brief history of neuroscience technology. So we go back to the 1930s. This was an era of single neuron neuroscience, and these neurons were ex vivo. So Hodgkin and Huxley are famous for their experiments that won them the Nobel Prize. The voltage probes were much bigger then than they are now, so they needed to find a really large neuron to stick the probe into, and they did so by finding the giant squid axon. And their success led to a famous circuit model called the Hodgkin and Huxley model. But if we fast forward to today, we've moved far beyond the single neurons and the ex vivo era. Now we have this remarkable ability to manipulate neurons in vivo. Modern technology allows all optical read-write capability using what's known as calcium imaging and optogenetic stimulation. We have millisecond precision. For example, if I plot for you the voltage of a neuron across time, as we stimulate, you see a peak in the voltage. That's called an action potential or a spike, or the neuron fires. And if you do that multiple times, we have millisecond precision and control over that. We also have single neuron selectivity. So here's 5 example neurons in vivo, and we're plotting the firing rate of those 5 neurons. The firing rate is the rate at which spikes occur. And they have some baseline firing rate. And then we have the control to stimulate one or multiple of those neurons. And we see that the firing rate then increases when we stimulate those neurons, but not the other neurons. We also are in this era of large scale in the number of neurons that we can measure. So with NeuroPixel technology, that's a probe that has about 1,000 voltage channels along it. And if you stick that into the mouse brain, you can measure hundreds, thousands of neurons at any location in the brain. So plotting simultaneously recorded neurons over time, the doubling time has been every 6 or 7 years. And it places us firmly in this era of scale in neuroscience. So what can we do in this new era of scale? And today I'm going to share with you the 2 projects from my PhD, which leverage these new technologies. One will be in perception. We'll look at one brain region, mouse primary visual cortex. And I'll talk about 2 studies, one in optogenetic stimulation that leaves us with an open question, and one on the structure of spontaneous activity that gives us another question. And then we'll address those questions with a theory. And then in part 2 of the talk, I'll talk about decision-making. We're going to zoom out from a single region of the brain to brain-wide activity. And that is made possible through a collaboration called the International Brain Lab. I'll show you some decoding analysis, the technique we use to analyze the data, and the results that come out of that. To motivate the study on perception, I'd like you to consider this question. What's the dimmest light that you can perceive? So imagine you look at a bright star, photons enter your eye, the photons excite cells in your retina, and that passes the information to your brain, your thalamus, which routes the information to your primary visual cortex. Primary visual cortex is going to be the focus of this study. And to make this question a little bit more precise, I want to ask, what is the minimum amount of neural activity that you need in primary visual cortex to perceive light? So for example, if you look at a dimmer star, you have less activity in primary visual cortex, but you could still perceive it. You might say, yes, I still see it. But at some point, the star is so dim, the neural activity in primary visual cortex is so low, you cannot see it. And we would like to have fine control over the activity of primary visual cortex. And we can do that instead of showing you a star, we could actually stimulate the neurons in that region of the brain. We stimulate a number of neurons and decrease that number of neurons until you can no longer perceive. And also, we don't want to do this in humans. So you're not going to say yes and no, but we'll train a mouse to lick and to not lick. And before going in,
Look at the stars.
yeah, I tied it in. So before giving too many details about the experiment, I need to describe an important feature of what we already know of primary visual cortex. And that important feature is called orientation tuning. If I show you a vertical grading, a certain set of neurons will fire in your primary visual cortex. By the way, primary visual cortex, we just say V1 for short. So vertical grading, a certain set of neurons fire. If I show a horizontal grading, a different set of neurons fire. And so this is called orientation tuning because the neurons are tuned to a certain orientation of grading. It doesn't have to be just vertical and horizontal, but this applies for all angles of gradings. So knowing that, we can now be more clear about our hypothesis. It is that direct stimulation of a small number of similarly tuned neurons can trigger perceptually guided behavior. To address that hypothesis, we formed a collaboration between experimentalists and theorists. On the experimental side, I want to highlight James Marshall, Timothy Mikado, and Sean Quirin. They were in Carl Diceroth's lab. And on the theory side was myself working with Jonathan Cadman in Surya Ganguly's lab. Here's the task that the mouse was trained to do. The mouse is shown the vertical and horizontal bars. There's different contrasts, but you don't have to worry about that for now. When it's shown the vertical grading, it's supposed to lick. And when it's shown the horizontal grading, it is not supposed to lick. If it does this correctly, it's given a water reward. And if it messes it up, it's given an air puff. All right, so now the mouse, as it's trained to do that, it's head fixed, and we're performing calcium imaging. So we're looking at the neurons as the mouse is performing this. So we can identify the neurons that fire when the vertical grading is given. So we see those green neurons firing, and then the mouse licks. What that allows us to do is we can remove the stimulus altogether and just stimulate those neurons using optogenetic stimulation. And as you might expect, the mouse licks. So now we're set up, we can decrease the number of neurons that are stimulated until the mouse no longer licks. When we do that, we find that a surprisingly small number of stimulated neurons can drive behavior. So if we plot neurons stimulated versus the percent correct behavior, we see it only takes 20 neurons to reliably drive behavior. We can also ask, what is happening to the similarly tuned neurons in V1? So by that, I mean the other neurons with the same orientation tuning. So I plot the fraction of similarly tuned neurons that are activated when we stimulate against the number of neurons we stimulate. And we find that this is a large fraction. 20 to 30% of neurons are driven when we stimulate. And the total number of neurons that are similarly tuned can be in the thousands. And so this large fraction, what this means is that tens of neurons can drive activity in thousands of neurons. And that's going to be a key puzzle for us today. By the way, if you stimulate 0 neurons, in other words, spontaneous activity, you see a very low rate of response. So somehow spontaneous activity is stable. And yet, when you give it this small kick of stimulation, you're able to drive large activity. How is that possible? Is there something special about the structure of spontaneous activity that would allow this to occur? Here's an example of spontaneous spiking activity from mouse barrel cortex from Mariani et al. The plot shows neuron index by time, and there's a black tick where every spike occurs. Mariani et al. Analyzes the structure of spontaneous activity by defining avalanches. And avalanches are patterns of neural activity that are highlighted in gray on the plot. So they bin all the neuron spikes in time, and an avalanche is defined to be a series of non-empty bins which are bookended by empty bins. The size of the avalanche, which we call S, is the number of spikes, and the duration of the avalanche is proportional to the number of bins, that's called T. And the interesting thing that we find is if you look at lots of spontaneous activity, and you get lots of sizes, lots of durations, you look at the distribution of those sizes and durations, they tend to follow power laws, meaning P of S is proportional to S to some power, which we call minus alpha. And P of T is proportional to T to some power, which we call minus tau. So Mariani et al. Plot the distribution of avalanches and sizes, and they fit the power laws to find an exponent. They find that alpha equals 2.24 and tau equals 2.52. Another study, Fontenelle et al., takes the same analysis of avalanches, the same analysis of extracting alpha and tau, but they look at that across studies, across mice, rats, monkeys, and turtles. And if you plot these exponents alpha and tau that you get from the neural activity in all these cases, you find that the exponents tend to lie along this dotted line. And why is that? There's a couple studies that have tried to answer that. One of them is Porta et al. And they explain this dotted line with a model called the C-Ross model, where they tune it to the edge of a phase transition where oscillations occur. And near that phase transition, when they compute avalanches, they see power laws which have exponents that lie along this dotted line. However, to our knowledge, there's still no theory connecting this dotted line of exponents with a phase transition. So we'd like to develop a theory. Just to step back then, we'd like to develop, it'd be really nice to have a single theory that explains both of the question, both the high sensitivity from the first experiment and the spontaneous avalanche exponents from the second. So before exploring the full neural network model that we cook up to try to explain this, I need to explain to you the single neuron model. If you remember back to the first slide, Hodgkin and Huxley developed this single neuron model using the giant squid axon. And the circuit looks like that. We actually don't keep any of the components except for a resistor and a capacitor. We keep some things for the basic physics, but we do remove most of it to keep the network analysis tractable. So now what we have as a model of the neuron's membrane is just a resistor and capacitor in parallel. From the outside of the membrane, we have input currents that are spikes from other neurons. And the variable that we're really interested in is the voltage difference between the outside and inside of the neuron membrane. We call that V. And we can solve for the evolution of V by solving the circuit. So we have this blue equation for V dot. And then we model The input currents, again, is this spikes from other neurons. So this is the spike train multiplied by some constant J. When the voltage is integrating in the blue equation, when it gets up to a threshold theta of 20, then it spikes. And it spikes, at theta. Here's an example of this. So I plot the blue equation. We plot the voltage integrating in time. And the input spikes are in orange. So you can see as a spike comes in, the voltage goes up, and it keeps going up until it gets to 20. And if there's a spike, which we signify by the red dot, then when a spike occurs, the neuron resets, which means the voltage goes to 0. And it stays there for a refractory period, which we call tau refractory, and that's 2 milliseconds. After that, it continues to integrate as it did before. That's our single neuron model. Now we want to connect lots of neurons. We connect N of these single neurons together into a network model. This is known as Brunel's model, which is over 20 years old. Each neuron has an index I, and the voltage of that neuron is described by this equation from the single neuron model. But now there are some key differences. Wij defines how all the neurons are connected to each other. D is a spike transmission delay. W external defines the contribution of an external population of neurons. And these neurons have spike trains, which we call SK. Let's walk through the connectivity matrix. Pictorially, we have an excitatory population that excites itself and an inhibitory population. And we have an inhibitory population that inhibits itself and the excitatory population. Mathematically, Wij has a 2x2 block structure where the excitatory blocks have a positive constant J and the inhibitory blocks have a constant minus G times J. We define J to be 0.1 millivolts, and the ratio of the number of inhibitory neurons Ni to the number of excitatory neurons Ne is fixed at 1/4. That's motivated by the ratio that's seen in cortex. And then this matrix is sparse. So we do an element-wise multiplication by a sparse binary matrix. The rows of this matrix have exactly 320 non-zero elements for the excitatory population, and then 80 non-zero elements for the inhibitory population. Now we can look at W external. There's this external population that has an excitatory drive to both the E and the I networks. All the neurons in the excitatory population, all the neurons in the external population are excitatory. So they have this positive constant J. Again, we multiply by the element-wise matrix, multiply element-wise by a sparse binary matrix. The number of non-zero elements per row is 320. We define the spike delay to be 1.5 milliseconds. And the external spike trains, we just take to be drawn from a Poisson process with firing rate nu external. So this is the network model, but what should you remember from this? You should remember 2 parameters. Please remember G, which is the inhibition dominance. So if G is large, then we have strong inhibition. You can expect to see low firing rates in the network. If G is low, you have weak inhibition, excitation might dominate, and you could see large firing rates in the network. The other variable that you should remember is called V. V is the non-dimensionalized version of nu external. So V is the external excitation strength. And if that is high, you have a strong excitatory drive, you can expect higher firing rates. And if it's low, you expect lower firing rates. So now we have G and V. We can change G and V and look at what happens to our model. This means, so we choose a model of size n equals 320,000. So that means there's 320,000 of these equations that we want to solve simultaneously. We do that with Euler integration. How can we visualize the network activity? One way to do this would be to show you all the spike times of the neurons. So here are some example points in this phase diagram of V and G. And if I plot the neuron index versus time, and I put a tick where there's a spike, you can see in blue, the external excitation is very low, so we don't have much network activity going on, low firing rate. But if we increase the external excitation just a little bit and go to the orange point, now we see these interesting sharp events that seem to occur. If we increase the inhibition dominance in red, well, initially we have one of these sharp events, but then the system relaxes down into what might be a fixed point of some kind. If we decrease inhibition dominance at the green point, we see these sharp events come back. How can we get a better intuition for what's going on here? And physicists love to coarse grain systems. One way to coarse grain the system is to define a population firing rate. That's the rate of spikes that occur across the whole population divided by the size of the population. So here are population firing rates on a log scale, and they show a similar story to what we saw with the spikes. But a variable that's more fundamental to the equations is V, because V is the variable that we're integrating. So in the same spirit of coarse graining, we'd like to look at the membrane voltage. We can plot the distribution of the membrane voltages across the whole network. And this distribution will evolve in time. So let me give you an example. At the blue point, click this. Here's a video of the membrane distribution. We have a bump of voltages that increase towards the threshold of 20, but they never get to the threshold. They stay just below it. So you have very low activity of the network. But if we increase the external excitation a little bit at the orange point, it looks like we have the same story, but now there's just a little bit more excitation, so you get to threshold. And then all the neurons fire, they reset to 0, and then they slowly come back. And this will continue to happen. If we kept watching the video, that cycle would continue to occur. So it really looks like we have a limit cycle here. If we increase inhibition and look at the red point, it starts to look the same. Maybe we'll have a limit cycle. But the inhibition relaxes the system enough that we get to what looks like a steady state, a fixed point in the.
Before, was it relatively periodic, or it was sort of bursty? You said it's a limit cycle. I think it'd been.
Yeah, if we kept watching that video, the bump would keep going to 20 and reset to 0.
But periodically, rather than almost stochastically.
Periodically, yeah. There's a pretty fixed time between events. And if we decrease inhibition to the green point, the limit cycle returns. If we kept watching this video, we'd see the limit cycle continue. And yeah, there's a characteristic frequency of that cycle. How could we come up with a theory for this voltage distribution? It'd be really nice to have an equation rather than showing you the simulations. The key to a theory would be ignoring the labels of the neurons, so that the only distinguishing feature of the neuron is the voltage. Here are the equations for all the neurons, 1, 2, I through N. We'd like all of these equations to be the same so that we could put them into a voltage distribution, but the boxes are not the same. The boxes have structure that depends on W and W external, and it depends on the specific timing of the spikes. Now, if the structure of W is disordered enough, and if these spike trains are uncorrelated, at least beyond the population firing rate, we could hope to approximate these boxes with a sum of Poisson processes. That's a big jump, but this is a mean field approximation. So This sum of Poisson processes is called the mean field term. And now all the equations are the same. So we can ignore the labels and put them into a voltage distribution. Mathematically, these are Langevin equations, and there's a standard process to convert that into a master equation or a Fokker-Planck equation, and that gives us the theory of P of V. So we have this mean field theory, and we apply dynamical system analysis. Between the red and green points, we saw that limit cycle appear. We find a Hopf bifurcation. And between the blue and orange points, where we also saw the limit cycle appear, we see a saddle node bifurcation. But that's a little odd for a saddle node bifurcation. We wouldn't expect a limit cycle to occur on a saddle node. So let's dig into the theory around this saddle node. So we're going to look at, on the phase diagram, this orange point right above the saddle node. We have this theory, P of V, that's also a function of time. For that theory, we can calculate a fixed point. The fixed point is this black dotted line. And we can also show the dynamics. So we can initialize the theory at time 0. We can initialize it at that fixed point. So red is time 0. And then as time goes on, the colors change towards blue. So you see a bump appear, and that bump moves around. Clearly, this fixed point is not stable because we initialized it there and something different is happening. But it's hard to tell on this plot maybe. So it'll be nice to look at it in a reduced dimensional space. If we instead of that plot a 2D space where we plot mean voltage versus the firing rate, this time the fixed point, that black dotted line, is represented by this blue dot. And the dynamics are the colored X's that go from red to blue. So we initialize it near that fixed point. The bump appears and moves upwards towards the threshold of 20. And then when it's near threshold, everything fires like we saw in the video. When that happens, the voltage goes towards 0 because the neurons are resetting. So we go down. And then slowly the voltage increases, the firing rate decreases and the voltage increases. And we see that bump from the video moving up towards threshold again. And if we keep watching the video, we would keep going around it. So this is nice because it reveals what is a limit cycle, this circle we keep going around. What happens when we go just below the saddle node bifurcation? So this time in the theory, we have 3 fixed points we can solve for. There's a low, middle, and high fixed point. The low one is this black dashed line. The middle one is the dot dashed line. And the high one is the dotted line. If we initialize the system at the middle fixed point, we see that's unstable because the voltage distribution moves away from that. And eventually, at large times, T of 300, the system settles into the low fixed point. It's nice to see this on our 2-dimensional space where the low, medium, and high fixed points are blue, orange, and green. So when we initialize at the middle fixed point, it's unstable. And it looks like we're going around the limit cycle from the last slide. But now we get stuck at this fixed point in the bottom. And if we think back to the optogenetic experiment where we're trying to explain high sensitivity, this looks promising. What if we simulated a system at this blue point and then gave it just a small kick? Maybe we stimulate tens of neurons. If that kick is enough to get it to this unstable fixed point and to go all the way around the limit cycle, now with tens of neurons, we've excited a network-wide event, or this might be thousands of neurons firing. So let's take a look at some simulations to see what happens. Here I'm going to simulate in the phase diagram. In the bottom left, there was a blue point. Now it's red, and now I call it AII. So sorry for the switch there. But at that point AII, we simulate the network. We stimulate some number of neurons, By the way, the network size is 4,000 neurons. We stimulate some number of neurons, and we plot the probability of a large response occurring. If we stimulate 0 neurons, the spontaneous rate of these large responses is very low. So that's good. That's like what we saw in the experiment. Then if we increase the number of neurons we stimulate to 40 or 50 neurons, now we have a good chance of driving a large response. So tens of neurons are driving activity and thousands of neurons. We'd like to turn to our theory now and ask, what does our mean field theory tell us about this kind of stimulation? We can include a stimulation term in our mean field theory. Specifically, we define a stimulation pulse, where we stimulate the excitatory population at a rate nu stim for a duration of sigma equals 300 milliseconds. We do this in both the simulations and the theory and plot the population firing rate over time. In blue, the stimulation size is small. The baseline firing rate is low. And when the stimulation comes on at time 0, not much happens. But in orange, the stimulation size is just large enough to drive a large event. And if we continue increasing stimulation size in green, we see multiple large events occurring. And black is the theory, which matches the simulations well. Now we can define the mean response of the excitatory population, and we call that delta nu. As we increase the stimulation size, nu stim, we plot the probability of a stimulation given delta nu. So that gives a heat map, which is in red. And this heat map shows these discrete steps, which correspond to the discrete number of large events that are driven by the stimulus. If we plot the theory in gray, it explains these steps well. And the colored dotted lines are the colors corresponding to the stimulus sizes on the left. We can also do this at multiple points in the phase diagram, A, B, C, and D. At A and B, we see A and B are near the saddle node bifurcation, which is responsible for these discrete steps. And we see the discrete steps at A and B. We see it partially at C, but if we look at D, for example, which is near the hop bifurcation, we no longer see those discrete steps. So the saddle node bifurcation is very important to our theory. It's important to answering this sensitivity question. And indeed, we see this high sensitivity. So is the saddle node bifurcation also going to help us to answer the question about avalanches? So we use our theory near the saddle node bifurcation, and we start by defining firing rates. We plot the firing rate R for both simulations in green and theory in gray. Here, I'm showing you the green point in the phase diagram. You could do the following with the other points on the phase diagram, but green is the clearest example. So these firing rates vary in time, and we can define a distribution of firing rates W. And here we've divided the firing rate R by the mean firing rate, R bar, and that's to be consistent with how avalanches are analyzed. There's a theory developed by Priestmont et al. That's based on inhomogeneous Poisson processes, and it allows us to convert this rate distribution into avalanche distributions. So in green, we plot the avalanche distributions of our theory, both for size and duration. And in gray, we plot the avalanche distributions from our theory. Green is simulations, gray is theory. And we find that they agree well. We can fit our simulations to power laws and extract the exponents, alpha and tau, that you might remember from the beginning of the talk. The simulations do lie along that dotted line that we'd like to explain. Can we use our theory connected with the saddle node bifurcation to explain this dotted line? The saddle node bifurcation affects the distribution of rates W, by introducing large and rare events. This means that near the saddle node, large firing rates have non-zero values in the rate distribution. So we can parameterize a family of rate distributions that are consistent with that feature of the saddle node bifurcation. The family of rate distributions produces a family of avalanches, and we can fit those to find a family of alpha and taus. And that family of alpha and taus describes well the dotted line that we see in the literature. So to step back, I'll summarize part one of the talk. We talked about an optogenetic study, which found high sensitivity. We talked about the structure of spontaneous activity. We had an unanswered question about avalanches, and we addressed both of those questions with a mean field theory of leaky integrate and fire neurons that's poised near a saddle node bifurcation. All right, so now I'll move on to the second part of the talk. The first part of the talk was motivated, at least in part, by one region of the brain, primary visual cortex. Now, I'd like you to zoom out with me to the whole brain. Are there some phenomena that require broad distributions of activity in the brain? Well, yes, decision-making is one example of such a phenomenon that requires brain-wide activity. So we'll zoom out and study decision-making. This is possible through a collaboration called the International Brain Lab. I'll talk about the analysis we use to describe this unprecedentedly large data set. And then applying that analysis to the data set, I'll show you the results. So how does neural activity in hundreds of brain regions give rise to decision-making? And understandably, it's been difficult to answer this question. And that's because studies differ in various ways. For example, different labs are doing the experiments. They look at different regions of the brain while the animals are doing different behaviors, and they may use different analysis methods. It isn't surprising that it's hard to find some unified conclusions about decision-making. In order to solve those problems, we study decision-making on a global brain scale in mice using multi-region neural pixel recordings. And to do this, I work with a collaboration called the International Brain Lab, or the IBL for short. The IBL is a collaboration of 11 experimental labs working together. Each lab collects data using neuropixel probes, which have 1,000 voltage channels along them, and you might remember that technology from earlier in the talk. These probes can be inserted into the mouse brain to measure in vivo neural activity. Different labs have inserted these neuropixels at different locations in the brain, and then the data is pooled into one data set. Normally, we should be skeptical about pooling data across labs, but the IBL has produced papers that show both behavioral and electrophysiological reproducibility across labs. So here it's okay to pool the data, and doing so yields 547 insertions across 115 mice and 300,000 units of neuron-like activity. If you view all these insertions in a graphic, you get these fun brains that I like to call porcupine brains.
Sorry, what is a unit?
A unit, so it comes out of spike sorting. It's like a neuron. I just don't want to call it a neuron because it could be multiple neurons technically. With this data, we can study 123 regions of the brain, but it's hard to visualize those in 3 dimensions. So we use something called a Swanson flat map, which flattens the 3D brain and displays it in 2 dimensions, and it looks like this. So you'll see some more of these in my next couple slides. The basic analysis of this data set is presented in our paper, a brain-wide map of neural activity during complex behavior. This paper is in prep, and we plan to release it on preprint soon. I want to highlight those that I worked with most closely for the decoding analysis. Those were professors, Peter Diane and Alex Pouget, and fellow researchers, Matthew Whiteway, Bert Gersick, Charles Finling, Felix Hubert, and software developer, Julia Huntenberg. And I will spoil the ending for you. We do find variables related to decision-making in over 100 regions of the brain. But you'll have to stay tuned to see those results. First, I need to describe the mouse task. The mouse is in front of a screen where the visual stimulus appears either on the right or on the left. The mouse needs to make a choice. It needs to turn a wheel in order to move the stimulus into the center of the screen. If it does so correctly, it gets a water reward. And if it does it incorrectly, it gets a noise burst. Another important feature of this task is that the mouse has to use its prior beliefs about where the stimulus will appear. That's because the contrast level changes, and sometimes the contrast level is 0. If the stimulus has 0 contrast, then of course the mouse cannot see it, and it has to make a guess using only its prior beliefs. In order to study the prior belief, we introduce what is called a block. A block is a series of consecutive trials with the same probability of a rightward stimulus. So if I plot the probability of a right stimulus, this is a percentage. The first 90 trials, we always do 50% probability, but then we start blocks. So we might go into a right block, which is the blue strip. So for some number of trials, the probability of a right stimulus is 80%. Then we have a stochastic transition to a new block. This is called a left block. So the probability of a rightward stimulus is 20%. And then after a few trials, there's a stochastic transition back to the right block. So we have these 2 blocks, right block and left block, and we stochastically transition between them throughout a whole session. How do you know if the mouse is using this block prior? Well, if we plot the mouse's performance on 0 contrast trials against trials relative to the block reversal, we see that the mouse's performance dips right when a new block comes on. And again, these are 0 contrast trials. So the mouse can only get it right if it has the right prior belief about where the stimulus will appear. Now, even though it gets it wrong right away, after 5 or 10 trials, the performance increases above chance level, indicating that the mouse's prior beliefs align with the block. Now that we've defined this task, I want to move on to discuss the analysis that we use. This is called decoding analysis. Here's some example neural data that's been aligned to different task variables. From the top to bottom, we have the block side, which is either right or left, the stimulus side, which is either right or left, the choice, which is right or left. We have the wheel velocity, which is a continuous variable. It's how the mouse moves the wheel. Then we also have the mouse whisking, motion energy of the whisking, and licking. Then we have neural data. The neural pixel probe is inserted in this example such that it goes through midbrain and hindbrain. And we show you the spike sorted data here. And it's 3 example trials, 242 to 244. Decoding analysis, simply put, is regressing the target variables from the neural data. So let me define the regressors first from the neural data. We define this trial relative window. For example, for block, the trial relative window is -400 to -100 milliseconds relative to stimulus onset. And these gray boxes are examples of where those would fall in the trials. And then we sum up the number of spikes in those trial relative windows for each neuron and for each trial. So this gives us a matrix X, which is neurons by trials. And these are our regressors. We can also do this for other variables and other trial relative windows. For example, stimulus, we're interested in right after the stimulus comes on. So we use a trial relative window, which is 0 to 100 milliseconds from stimulus onset. For the choice variable, the trial relative window is -100 to 0 milliseconds relative to the movement onset. And for feedback, it's 0 to 200 milliseconds relative to feedback onset. Now we want to define the target variable of our regression. So we choose a target variable. That target variable should be chosen to be the same as the variable we use to define the trial relative window. For block, stimulus, choice, and feedback, they're binary, they're right or left decisions. So we can just define those to be 0 and 1. So y is this binary vector of length trials. And we want to perform regression to predict y from x. And we do regularized, cross-validated logistic regression to do this prediction. Here's an example of the regression from the regression of block from the region caudoputamen, or CP. The target variable is in black, and the predictions are in purple. We quantify the decoding performance using an accuracy which is balanced across classes. So for an accuracy, we would expect chance level to be around 0.5. So is this balanced accuracy good, 0.74? Well, we might think it's good because it's higher than 0.5, but we can't do that because the block variable and the neural data have temporal autocorrelations. This means that each trial is not statistically independent from the rest, and it makes decoding performance better than we would expect from traditional intuition. Let me give you an example. The block is a stochastic variable, so we can simply resample the block variable, and we call this resampling a pseudo session. So in blue, orange, and green, I show 3 examples of block pseudo sessions, and then we decode those pseudo sessions. And our decoding scores, the balanced accuracies, are better than chance. They're 0.66, 0.61, and 0.56. But remember, the mouse never saw these pseudo sessions. We just generated them from the same generation process as the real one. So we can do this same process instead of 3 times, we could do it 1,000 times. And we can plot the distribution of pseudo session balanced accuracies that we get, and that's in gray. And you can see that almost all of them are above the chance level of 0.5. So again, we don't want to be comparing against 0.5. We want to be comparing against this, which we call the null distribution. We define the gray line to be the median of that distribution, and the purple line is the real decoding balanced accuracy from the purple predictions. Significance is going to be defined relative to this gray distribution, and the effect size will define to be the purple line minus the black line. So now that we've gone over the task and the analysis, I could start to share the results with you. And the specific question that we'll be asking is, which brain regions can significantly decode stimulus, choice, feedback, and block? We're going to go through these variables one at a time, and I'll start with stimulus. Stimulus side decoding is significant in 28 out of 123 regions. The bar plot shows the 28 regions, which are significant. And the ordering of the bar plot and the colors is actually important. So I've added a key in the bottom left so you can remember. The cortical regions are first, and they're in green. Striatum is next, that's in blue. Thalamus is in pink. Midbrain is in dark pink and orange. And hindbrain is in light pink. Also, cerebellum, you can't see, cerebellum is in yellow. Cerebellum isn't on this plot because there happens to be no significant regions in cerebellum for this example. So in the bar plot, we show each region has multiple sessions of data. Each session gets a dot. So a black dot means that session is significant at alpha of 0.05, and an empty dot is insignificant. The bar height is the median balanced accuracy across sessions, and the white point captures the median of the null distributions. There are some areas that are in this plot that are classically considered to be visual areas. For example, there are visual cortex areas, visual thalamus areas, and superior colliculus. There are also other areas that we find. For experts, these may not be surprising, but they're not traditionally called visual areas. And these are striatum, midbrain, and hindbrain. And I'll highlight some regions of interest for the neuroscientists, though I admit if you haven't seen these acronyms before, this just looks like alphabet soup. So I'm sorry about that. Finally, I want to put these regions onto a Swanson flat map. This figure is taken directly from the brain-wide map paper, and the figure was generated by Christopher Langdon. The color of this Swanson flat map is simply because the color scheme of the paper is this way. It's green. The effect size of balanced accuracy over the null is shown as in the color bar. Gray regions are insignificant at alpha of 0.05, and white regions are not analyzed. So now I'll move on to another variable, the choice variable. We find significant regions are broadly distributed for choice. In 99 out of 123 regions, it's significant. Some areas that would be considered classical for choice decoding are cortex, hippocampus, striatum, and midbrain. We also find other areas in our study. Those include thalamus, hypothalamus, hindbrain, and cerebellum. By the way, here's this yellow that you couldn't see from the last slide. That's cerebellum. And I want to highlight some non-cortical regions that come up with especially strong decoding. Whoops. Yes, here's highlighting some regions. And here's the Swanson flat map, again, taken from the brain-wide map paper with the color scheme from the brain-wide map paper. And now we'll move on to the feedback variable. Feedback decoding is broadly distributed. In fact, it's almost everywhere. There's only one region that we don't find it in, and that is PA, which is posterior amygdala nucleus. Why is feedback everywhere? Well, there's a lot of factors to feedback. For example, there's the reward delivery. The valve makes a clicking noise when it releases water, so you would expect the mouse to hear that, and you could decode that. The mouse has to consume the water, so it's licking. And there's strong neural signal for licking. There's an error tone when it gets it wrong, so that's a strong auditory cue. And we'd also expect there to be an effect of neuromodulatory systems, such as dopamine, which has very widespread connections in both cortex and subcortex. So now I'll move from the variable with the most regions to the variable with the least regions, and that's the block. We find fewer significant regions in block than for stimulus, choice, and feedback. We find 3, and those are MOP, that's a premotor area, CP, that's in striatum, and PPN is in midbrain.
So could you say what the block means again?
Yeah, the block is a series of consecutive trials with the same probability of a rightward stimulus. So on the zero contrast trials, for the mouse to do better than chance, it needs to keep track of the block. This
is whether you can detect which block you're in by looking at these.
Yes, yep. So looking at this neural data, we can make significant predictions about which block we are in. The signal associated with the block prior may be more broadly distributed than just these 3 regions. For example, 15% of regions, or 19 of them out of 123, survive at an alpha of 0.05. And that includes regions in cortex, striatum, thalamus, midbrain, hindbrain, and cerebellum. They're not on this bar plot because we do a multiple comparisons correction, which is stricter than the alpha of 0.05. But if I show you all of the areas significant at alpha of 0.05, those 19 regions are on this Swanson flat. Another reason why the prior beliefs may be broadly distributed is because the mouse can't know the block exactly. The block is a stochastic variable controlled by the experimenter, but it could have some internal representation, which we call the subjective prior. So you could think of that as a smoothed out version of the block in time. And there's a companion paper to the brain-wide map paper, which studies that prior. And they find that it's more broadly distributed than just these three regions, but I'm not going to talk about that paper today. To wrap up this section of my talk, I introduced to you the International Brain Lab collaboration with these 11 experimental labs. I shared with you the decoding analysis, and we looked at the decoding results. We found various regions that decoded significantly for stimulus, choice, feedback, and block. This is the outline that I promised you we'd go over today. Part 1 on perception in one region of the brain. We developed a theory to address some questions found in experiments. And in part 2 was decision-making. We zoomed out to the whole brain. We looked at an unprecedented amount of neural activity, and we looked at the results of different variables and found different brain regions that were significant. Looking to the future, of course, there's still unanswered questions. In the perception study, we found V1 to be very sensitive. Are there other brain regions that are also as sensitive as V1? Regarding avalanches, is the saddle node bifurcation the only bifurcation that constrains avalanche exponents, or could there be other bifurcations that do the same? And in the decision-making study, where does the computation of the subjective prior happen? Does it happen in one region and it is then projected elsewhere? Or is it computed in many places? And finally, an education component to consider. The IBL is committed to open science, which means all of its data set is online and freely available, and all of its tools are available. The coding software is available. So how can we best disseminate that data set and tools and build the research capacities of universities that have limited resources across the world, and what that have an impact on education? So I have a couple acknowledgement slides. I want to thank the International Brain Lab so much. I can't highlight everybody. I'm sorry. There's a lot of people in the International Brain Lab. So I've just highlighted a few. But of the PIs, I worked closely with Peter, Diane, and Alex Pouget, and I'd like to thank them very much for their patience in working with me. I also have gotten help from Tatiana Engel, so I want to thank her for her career advice. And I've learned a lot of statistics from Kenneth Harris, so I want to thank him. Hannah Bayer is the fearless leader of the IBL. I thank her so much for her administration and leadership is amazing. And software developer Julia Huntenberg has worked with me a lot on the decoding process. There's also some researchers that I worked with most closely, and I'll highlight them and thank them. Michael Schartner, Matthew Whiteway, Ryland Schaefer, Bert Gersick, Charles Finling, Felix Hubert, Yanliang Shi, and Christopher Langdon. Thank you so much to all you IBL people and to our funders. I'd also like to thank the Surya Ganguly Lab, Neural Dynamics and Computation. Again, I can't highlight everyone. Thank you so much to all of you for being here and for sitting with me throughout all the years and struggles. Thanks to John for helping me with the spiking networks.
You're welcome.
And Ben, I highlighted because I got to TA with him this summer at a course that Steve Backus leads in Woods Hole, and that was very fun. And I worked with Jonathan Cadman on that part 1 of the talk, so I thank him. And thank you to funding. This picture was from 2019 in Woods Hole. This is the MCN course. And for some reason, Ganguly Gang ended up going there, meeting. I don't know why there's so many of us there, but yeah, we were all there. And thank you to my committee members. It was pretty cool. You were all here before 1 p.m. So that means a lot. Thank you, sorry. Janet Carlson, I just cold emailed you And you decided to be my chair, so I'm really thankful for that. Thank you, Ben Good. I got to take Daniel Fisher's class a few years ago, so thank you for that. And Steve Backus, you've given me great mentorship, career advice, and in the MCN course, I got to TA for that. That was really a pleasure and a joy. Thank you. And I need to thank the staff of the Applied Physics Department because none of this is possible without them. Claire Nicholas is so kind. Every time you sit down with her, you just feel loved and cared for. Thank you to Patrice O'Dwyer, Fiona Chu, Jessica Kubo, and Ingrid Tarian. Ingrid, thank you so much for making sure I always get reimbursed. You're amazing.